Today we will learn another bacteria which belongs to the group Enterobacteriaceae. This bacteria is named as Klebsiella. Now, a little bit about the history. One of the first pioneers was Carl Friedlander. He first described this bacteria in patients who died of pneumonia after performing autopsy on those patients. And in that, he was able to show that these were small bacilli which were capsulated. At that point of time, this bacteria was also called as Friedlander's bacilli. Thereafter, this bacteria is today what we know as is a gram-negative bacteria, which is known to have capsule. Furthermore, this is a non-motile bacteria and it belongs to the group Enterobacteriaceae. And among the group, it belongs to the tribe Klebsiella. Okay. Now, why this bacteria is important? You have to remember 2 points. First is this bacterium can survive in the environment. Furthermore, it also colonizes and is part of the commensal flora of the human beings. To add to that fact, this bacteria has multiple mechanisms by which it becomes resistant to many groups of antibiotics. As of today, it is one of the most resistant organisms. Okay. Now, in the tribe Klebsiella, we have two main groups, Klebsiella, Hafnia, Serratia. We will be discussing today about Klebsiella. This tribe differs from the other tribes of Enterobacteriaceae by the fact that they react positively to VP test and negatively to methyl red test. Among this, we have three subspecies of Klebsiella pneumoniae. The first one is a subspecies pneumonia, or very commonly called as Klebsiella pneumoniae. This bacterium is a notorious bacterium, commonly causes respiratory and urinary tract infections in community acquired infections, and in the hospital or healthcare associated settings, it can cause any kind of infections from sepsis to surgical site infections to ventilator associated pneumonia. Name any type of healthcare associated infections, this bacterium will be associated. Two subspecies of Klebsiella pneumoniae are Klebsiella ozeni and Klebsiella rhinoscleromatis. These two cause two different groups of diseases. The first one is associated with atrophic rhinitis, and the second one is a possible etiological agent of rhinoscleroma of the nose. Today we will be focusing more on the Klebsiella pneumoniae subspecies pneumonia. When this organism is cultured on a routine media like a MacConkey agar, this is a lactose fermenting bacterium. Hence, it produces pink colored colonies. If you look at this picture, you will find on the MacConkey agar there is a luxuriant growth of pink colored colonies, and on the right hand side picture you can even see that the colonies are mucoid in appearance. That means it appears as if these are wet colonies, not dry colonies. This is probably attributed to the presence of capsule of the bacteria. Again, another picture showing the mucoid type of colony, and on blood agar we can find opaque pale white colored colonies. Okay. As far as the biochemical reactions are concerned, we need to differentiate between another lactose fermenting organism very commonly associated with human disease, that is the Escherichia coli. Now, if you remember from the last class, Escherichia coli is also a lactose fermenting bacteria. So obviously we cannot differentiate between them on the MacConkey agar. So we need to differentiate them based on the biochemicals. So what are the common biochemicals that were used? First is the TSI, or the triple sugar iron medium. In the last class I have told you, because they are both lactose fermenting, they will also ferment glucose and lactose. So in this case scenario, you will find that both Klebsiella and E. coli will have the same reaction on the TSI media, that is acidic by acid. The slant and the bud both will be yellowish in color, because both of them can ferment both glucose and lactose. So we will not be find any difference in that. The next comes the test, which is the indole test. What is the indole test? Anybody remember from the last class? In the indole test, if the organism has a particular enzyme called tryptophanase, it will degrade tryptophan to form indole derivatives. That can be detected. Now that reaction is positive in case of Escherichia coli, but is negative for Klebsiella. So this is one of the reactions by which we can differentiate between E. coli and Klebsiella. If you look at this picture, Klebsiella will give an indole negative reaction, whereas E. coli will give an indole positive reaction, which we can observe by formation of a red color ring at the surface of the two reagents. The bottom one is the liquid culture media, in which the tryptophan is degraded to form indole derivatives, and it is detected by the indole reagent. That is the first point of differentiation. The second point of differentiation will be methyl red. I already told you the Klebsiella group, the tribe of Klebsiella are negative for methyl red, but Escherichia tribe are positive for methyl red. Similarly, E. coli will give a positive reaction for methyl red, and Klebsiella will give a negative reaction. Next is VP test, which will be opposite. That means in case of E. coli it will be negative, in case of Klebsiella it will be positive. Finally, citrate. In the last class I already told you we are looking for citrate utilization. That means this media does not contain any sugar. The only carbon source is citrate. Some organisms can utilize this basic source of carbon, some can't. E. coli cannot survive only on this citrate, whereas Klebsiella can survive. Hence, when we do test, we can find that we will say the citrate is utilized by Klebsiella, as evident by change in the pH and development of a bluish color. And in case of E. coli, citrate will not be utilized and the reaction will be negative, that is green in color. Finally, urease test. What is urease test? I have already told you in last class. Some organisms are able to produce urease enzyme. This urease enzyme will break down the urea to form ammonium. This ammonium will change the pH of the color. Hence, you can see these two, like Proteus. We will talk about Proteus in our later classes. Proteus does produce more urease enzyme. Hence, the reaction is the change of pH is more. In case of Klebsiella, the change of pH is slightly lesser, but it is still positive for urease. So these are the differentiating points between E. coli and Klebsiella. So whenever you are given an organism in your examination, first look at the culture media, then try to describe the colonies. Okay. Now what can anybody tell how we do describe a colony on a culture plate? How will you describe? Okay. Now let us look at this. Suppose this is the culture plate you have given in your examination. So how will you proceed? What would be the first step? Yes? right? The first step is you have to identify the media. It could be a nutrient media, it could be a blood agar. First identify what is the media. Suppose you have been given this agar. So first we have to say this is an MacConkey agar plate. Next, what we see, we have seen there is a growth of bacteria. Bacterial growth is seen. Then look at the single isolated colonies. Then we start the proper description of the colonies. What would be the points by which we want to describe them? First would be, what is the size of the colony? It's a big, is it small? No, it's big colonies. You can state approximate size, only approximate, you don't need to get it exact, okay? You can say we have colonies about 5.5 mm diameter colonies. After size, next will be shape. Over here, what is the shape of the colony? These are circular colony, round. Some organisms will have stellate shape or no specific shape altogether. But over here, we are getting a proper shape. First would be size, then shape. Next, we have to look at the edges. Over here, all the colonies have a smooth edge. The edges are smooth. In some organisms, it will be irregular edges. Then comes color. What is the color? Over here, the color is pink. Now, because the agar is MacConkey, the pink color denotes it is a lactose fermenting colonies. Okay. And like Klebsiella, we are observing the colonies appear to be mucoid or slightly watery. You can say it is mucoid type of colony. That is how we have to describe. Okay. Now, after describing the colonies on MacConkey agar, what will we say? We have a lactose fermenting organism on the culture plate. What would be the next obvious step? You have to perform a gram stain smear, and on gram stain you have to observe what you will observe, whether it's a gram positive or gram negative cocci or bacilli. Suppose you have observed a gram negative bacilli, then what will you say? This given organism is a gram negative bacilli. It is a lactose fermenting organism, right? Then we need to differentiate between the common organisms. For example, the same feature will be seen also in case of E. coli and in case of Klebsiella, right? So how can we differentiate? By using the biochemical test, the one we have described. Furthermore, a very important differentiating point would be, like in the last class I have shown you, E. coli is motile. You all remember on hanging dog preparation? Now Klebsiella is a non-motile organism. So even based on motility, we can differentiate. Okay. So these are the many points by which we can differentiate. Now the main question is why I am teaching you all this, because in examination the examiner might ask you, if it is a gram negative bacilli, how will you differentiate between E. coli or Klebsiella? Your answer would be, we will start with motility testing. E. coli will be motile, Klebsiella will be non-motile. Further, we can confirm with the use of biochemical reactions. For example, E. coli would be positive for indole test, Klebsiella will be negative for indole test, and the others. Okay. Now, another important feature is the kind of diseases this causes. As the name suggests, Klebsiella pneumonia is a very well-known organism causing community-acquired pneumonia. And sometimes the patients will cough out the expectorant and it looks like this picture, reddish colored sputum. This is also compared with red currant jelly. Red currant is a flavor of jelly, so almost it has a jelly like consistency and red in color. So Earlier, the typical description was people will expectorate out, and the expectorant will look like red currant jelly. Furthermore, like in this chest x-rays, you can see it causes classical pneumonia affecting the lower lobes of the lungs, and we go through all the stages of pneumonia. You have been taught pneumonia in pathology? No, not yet. Next, this organism causes plethora of infections. Right from, look at the head, it can cause meningitis, it can cause infections in the eye like endophthalmitis, any type of wound infection, traumatic wound infection or a surgical site wound infection. Then it can cause organ infections in the organ and abscesses like hepatic abscess, pneumonia. It can also seep from blood into bones, causing osteomyelitis. And because it can gain access into the blood, it can also cause sepsis. Very important cause of sepsis, especially in patients who have intravascular devices. Similarly, like E. coli, it can also cause urinary tract infections both in community and also in the hospital settings. A capsule is one of the very important virulent factors of this organism by which it can resist opsonization by macrophages and neutrophils. Hence, it survives and spreads very rapidly. Furthermore, originally this organism was more commonly seen to cause respiratory infections in alcoholics. A possible explanation is chronic ingestion of alcohol disturbs the intestinal microbial biases, thereby leading to the propagation of this organism. And from there, because alcoholics will get repeated aspiration of gastric fluid into the lungs, because obviously debilitation from alcohol, this organism will gain access into the lungs and hence causing severe respiratory infections. Another important feature that you should know is Klebsiella pneumoniae has a variant. Some of the variants of this organism are called as hypervirulent organisms. Now, why this is important? Just look at this picture. Classically, it is seen that the organism is first, human beings acquire the organism to the nasal route. From there, there is local colonization in the upper and oropharynx and the nasopharynx. Thereafter, it will slowly colonize the gut. And if there is some risk factor, it will gain access into the respiratory tract, thereby causing pneumonia. But the hypervirulent strain, it can cause pneumonia even before it gains access into the intestine. That means right from the stage of initial colonization in the upper respiratory tract, either oropharynx or the nasopharynx, from there onwards it can cause pulmonary infections. Okay, so what was earlier called as like debilitated people who are alcoholics or other debilitation due to other causes, people with repeated gastric reflux had more propensity to be affected by respiratory tract infection of this organism. That is fine, but if the strain is a hypervirulent strain, even without all those risk factors, people might be affected. There are some groups of organisms called as ESKAPE pathogen. ESKAPE is a mnemonic for these 5 organisms: Enterococcus faecium, Staphylococcus aureus, Klebsiella pneumoniae, Acinetobacter baumannii, Pseudomonas aeruginosa, and Enterobacter species. Now, what is seen is that, especially in the hospital settings, these 5 organisms in their own way have multiple methods of being resistant to many groups and classes of antibiotics. That means when you are dealing with patients with multiple infections, these organisms will be commonly multi-drug resistant. If you do recall, in the class when we discussed about Staphylococcus aureus, we discussed about MRSA strains, the VRSA strains, and how those are so difficult to treat. Similarly, Enterococcus faecium, very commonly found are vancomycin resistant enterococci or VRE. Those are very difficult to treat. Similarly, in case of Acinetobacter baumannii and Pseudomonas aeruginosa, the MDR strains are resistant to almost all classes of antibiotics. Okay, so among them is Klebsiella pneumoniae. Many times, when you will be observing the patients will be infected with a particular strain of Klebsiella pneumoniae, which will be resistant to almost all classes of antibiotics used for treatment. Now, why I am telling you all this antibiotic resistance today? I know you have not been taught about the antibiotics. We will discuss them when you have been taught in pharmacology. Otherwise, it is very difficult to understand how the resistant mechanism. I am only trying to brief a little bit about how this resistant mechanism works. So, briefly, if we try to describe, suppose this is a bacteria. How will any antibiotic affect? Any idea? How does antibiotic work? Good. So, it will lead to disruption of cell wall. Anything else? Any other way? Beta lactam. Okay, that's inhibition of cell wall synthesis. Anything else? Right. So one would be protein synthesis inhibitor. Then DNA alteration. Anything else? It can compete with the metabolites. That is the things that are required. Remember folate pathway? Last year, even bacteria requires that. So anything which competes with the same enzyme, that means it can metabolism might be inhibited either permanently or competitively. So it can an antimicrobial agent might inhibit the metabolism, DNA replication or translation or transcription by protein synthesis. Or the last one is directly acting on the cell itself, direct toxicity. These are the common methods by which any agent can act as a antibacterial agent. Now, whenever a bacteria is exposed to any antibiotic agent, it will fight to survive. Obviously, it wants to live on, right? So, what will it do? It will devise some ways by which it can circumvent the action of the antibiotics. How? For example, if it affects the protein synthesis or DNA synthesis, DNA replication, it will try to inhibit the uptake of the antibiotic molecules. If the molecule is not uptaken, it will not work. If the antibiotic is interfering with the metabolism, what it will do? It will start finding new pathways of metabolism so that it can circumvent that pathway. Understood? If it causes direct toxicity, it will try to cover itself with a protective covering. Okay. Now, if later on we will be discussing more about how bacteria become resistant to antibiotics, but what I am trying to stress upon, Klebsiella pneumoniae has many methods to do so. It can form capsule. Because the capsules are negatively charged, most of the antibiotics will not be able to penetrate into the bacteria. Their uptake will be reduced. It can change the metabolic pathway so that many antibiotics acting on metabolism will be altered. Okay, and obviously, because the capsule is present, direct action both by neutrophils or any other antibiotics will also be limited. And there are many methods of gene transfer. I think you have already been taught conjugation, transformation, transduction, right? Klebsiella also forms what is known as biofilm, that is, colonies of bacteria living together. And when they live together, they share this genetic data. It may be plasmid mediated, it may be chromosomal mediated from one bacterium to another. Hence, this genetic data of how to be resistant or how to circumvent the action of antibiotics is shared easily from one bacterium to the other in that biofilm. So after a short period of exposure, we do find that the same organism which was earlier sensitive to many groups of antibiotics will ultimately become resistant to all. This is the problem statement that we have right now. So what we can do? The best action would be, first is to prevent the infections happening. That would be the first step. How can we do so? In the hospital, the healthcare settings, we have to implement strict infection control policies so that even if one patient gets infected with these kind of organisms, that organism does not spread from that patient to other patients. Next important would be to recognize the clinical signs symptoms, use appropriate laboratory diagnostic method, and treat them with proper antibiotics. We'll talk about antimicrobial stewardship later on, with proper antibiotics based on the susceptibility testing. If we altogether fail to do so, these organisms will spread more and more rapidly, causing even serious infections. Okay, this is why we need to learn about this organism.